Hello everyone, welcome to Sri classes. So today we are going to discuss about molecular basis of inheritance. So before starting this topic, I want to tell you all that this is a very important topic because this is a common topic that you are going to get in both botany and zoology. And you can expect at least 9 to 10 questions in all your entrance examination like NEET, AIIMS. So you can expect 9 to 10 questions from this topic. So this topic is very important. And before starting this topic, let us understand the topic itself. What is the meaning of this topic? Molecular basis of inheritance. इस topic का मतलब क्या है? Okay. So if you can see, there are some words like molecular basis of inheritance. So what is inheritance? Our first question is, what is inheritance? Inheritance means the transmission of characters from parents to offspring. Like you might have seen that some people saying that ये तो अपने मम्मी पे गई है, ये तो अपने पापा पे गई है. So what does it mean? Okay, why they are saying so? Because there are some similarity, there are some resemblance between parents and offspring. Okay, so what is the reason? Why such resemblance? Why such similarity? What are the factors responsible for this resemblance, for this transmission of characters from parents to offspring? Or else we can also say what are the molecules responsible. Or to be more specific, I can say what are the biomolecules responsible for the inheritance, for the transmission of characters from parents to offspring. Although we are not exact copy of our parents, but still there are some similarity, there are some resemblance. So what are the biomolecules responsible for it? If you can recall, you have read about, there is a chapter in grade 10 named as biomolecules. So can anyone please tell me what are the things coming under biomolecules? If you can recall, we have already read about it. The biomolecules are protein, lipid, nucleic acid, carbohydrate. Okay, so in this topic, we are mostly going to focus on nucleic acid. So what are the different types of nucleic acid present in our body? Okay, and here you can see it is written structure of DNA. So DNA is what? DNA is a nucleic acid or it is what? Okay, so we are going to study about it. We are going to cover all these things in this particular topic. Okay, so let me start. Okay, so these are the topics that we are going to cover. First thing we need to cover is structure of DNA. And the second thing is experiment to prove DNA is the genetic material. What are the experiment to prove that DNA is the genetic material? Why RNA is not the genetic material but DNA is the genetic material? So what are the experiment to prove DNA is the genetic material? Then we are going to cover central dogma. Then we are going to cover DNA replication. Then the next thing is DNA transcription and the next thing is DNA translation. And at the end, we are going to cover lab operon. Okay, so today in today's topic, in today's class, we are going to cover what is the structure of DNA. So as you can see, we have already discussed about inheritance. What is inheritance? The same thing is written here. Inheritance is the transmission of genes or characters from parents to offspring. Now the question is, what are genes? What are genes? If I'm saying genes, what are genes? Where genes are present? You can simply remember it in a very simple description, in a very simple language, that genes are the functional unit of DNA. Or else you can see genes are the segment of DNA that contain the code. Genes are the segment of DNA that contain the code for a specific protein. It is the basic physical and functional unit of heredity. Okay. So now the question is, what is DNA? Here it is written genes are segment of DNA. So what is DNA? So DNA is deoxyribonucleic acid. Okay, so what are the two types of nucleic acid present in our body? DNA and RNA. What is DNA? DNA is deoxyribonucleic acid. Okay, if you can split the term, you will easily get an idea that what is DNA. Here it is written deoxyribonucleic acid. So no doubt DNA is a nucleic acid. And this deoxy and ribo means what? Deoxy means devoid of oxygen. There is no oxygen. Is it like there is no oxygen in the whole DNA? No, it is not like that. at some particular position. So we are going to study about it. So deoxy refers to the absence of oxygen or devoid of oxygen, and ribo means what? Ribo terms has derived from the sugar base like ribose. Okay, when I'm saying ribo, I'm referring to ribose sugar. Okay, and it is a pentose sugar. What do you mean by pentose sugar? Pentose means pentagon. Okay, there are five sides are there. If I'm saying hexose sugar, means 6 sides are there. Okay, so what do you mean by deoxyribonucleic acid? Deoxy refers to absence of oxygen or devoid of oxygen, and ribo terms has derived from the sugar base like ribose. And what is this DNA? I hope now you all have some idea like what is DNA. DNA is a deoxyribonucleic acid, which is a molecule that contains the instruction an organism need to develop, live, and reproduce. Okay, it consists of all the instructions. So you can say, suppose you want to make some biryani or any kind of dish you want to make, you need a recipe. Okay, you need a recipe for it. You need the whole book, recipe book, where the ingredients are written, the procedure is written. So DNA here is acting as what? It is acting as a recipe book. Okay, which is providing instruction, which is giving us some idea that what are the things we need to produce this thing. Okay, so we are going to discuss about it. For now, just remember what is inheritance, what are genes, what is DNA, and what is the full form of DNA. So these are some of the basic concepts that you all need to be clear about, okay, before starting this entire topic. Okay, so these things need to be very clear. Now let us move to the next part. So nucleic acid, as it is made up of nucleotide. Now we know that DNA is a nucleic acid. Okay, what is the full form of DNA? Deoxyribonucleic acid. Deoxy means devoid of oxygen. Ribo means we are referring to ribose sugar. No doubt it is a nucleic acid. Now the question is, what is the structure of DNA? DNA is made up of what? What are the constituents? Okay, so nucleic acid, as it is made up of nucleotide. So this nucleic acid is made up of what? Nucleotide. Again, the question is, what is nucleotide? So what are the nucleic acid present in living system? We have already discussed about it. DNA and RNA. DNA is deoxyribonucleic acid and RNA is ribonucleic acid. Here, what is the difference between these two? Can you guess it from the name? DNA means I'm saying deoxy means there is no oxygen. In RNA, I'm just saying ribonucleic acid. I'm not talking about oxygen. Okay, so you can see DNA is deoxyribonucleic acid and it acts as a genetic material in most of the organism. RNA is ribonucleic acid. It consists of oxygen and rest of the components are similar to that of DNA. So in next slide, we are going to study about what are the constituents of DNA. Okay, no doubt DNA is made up of nucleotide, but nucleotide is made up of what? Okay, so in next slide, we are going to study about it. Just remember one thing, if someone will ask you what is the difference between DNA and RNA, you can easily say that DNA, you can guess from the name, deoxy means there is no oxygen. At what position we are going to study about it. And in RNA that oxygen is there. And there is one more difference that we are going to encounter in our next slide. There is a difference in nitrogenous bases. Okay, so let us see what are the differences. As you can see this diagram, I think all of you know where DNA is present. In case you don't know, just have a look at this diagram. Everything will be clear, will be very clear. After looking at this diagram, everything will be very clear. So you can see the cell. Inside the cell, there is a nucleus. Inside the nucleus, what are present? Chromatin fiber. Chromatin fiber contains to form chromosome. Okay, so mostly they are present in this form only, in the form of chromatin fiber. If I'm going to talk about the genetic material, they are mostly present in this form only. They are present in the form of chromatin fiber. Okay, at some particular stage of cell division, they contains to form chromosome. They contains to form chromosome. And if someone will ask you where DNA is present, so DNA is present inside the chromosome. Okay, you can just have a look at this structure. I have taken it from this particular source. I have written it here. So DNA, you can see this DNA and these are the chromatin fiber and the chromatin fiber contains to form chromosome. So if someone will ask you where DNA is present, you can easily say it is present inside the chromosome. Okay, and where chromosome is present inside the nucleus. And where nucleus is present inside the cell. Okay, so cell, inside the cell there is nucleus. Inside the nucleus there is chromatin fiber which contains to form chromosome, and chromosome consists of what? DNA. And DNA consists of what? DNA consists of gene. What is gene? Gene is the functional unit of DNA. Why it is called the functional unit of DNA? It is called the functional unit of DNA because it is responsible for the transmission of characters from parents to offspring. Okay, so genes are responsible for the transmission of characters from parents to offspring. Okay, I hope it is clear. Now let us move to the next part, the DNA. So now you can see, let us study about the structure of DNA. So DNA is what? It is an acidic substance identified by Misser in 1869 named as nuclein. Okay, so if someone will ask you, what was the name of DNA earlier? Now it is called DNA, but earlier it was known as nuclein. Okay, and structure of DNA was given by Watson and Crick in the year 1953 based on X-ray diffraction data produced by Wilkins and Franklin. Okay, so Watson and Crick, they gave the structure of DNA, but how they give this structure? Like they must have read from somewhere. Okay, how they got the idea about the structure of DNA? They got this idea from the X-ray diffraction data produced by Wilkins and Franklin. Okay, so you need to remember the name of these scientists because you can expect the question like in OUAT or in some other entrance examination. You can expect that they can ask you in what year or which scientist gave the structure of DNA and all. So everything which are marked in red, these are very important, so you have to remember it. Okay, all those things which are marked in red, I have marked it in red due to some reason only. So you have to remember the name of this scientist, Watson and Crick in the year 1953, Wilkins and Franklin. Okay, what they did, okay, they were the reason for this X-ray diffraction data, and by referring to this X-ray diffraction data, by taking it as a reference, Watson and Crick gave this model structure of DNA. Okay, so according to them, what is the structure of DNA? So DNA is a double helix model. It's a double helical structure. So according to Watson and Crick, what kind of structure DNA is having? DNA is having a double helical structure. So if you want to see, you can see. Let me show you. So this is the structure of DNA. It's like a helix. Okay, as you can see, ये देख सकते हो, ये एक helix है, ये एक helix है. So दो helix है, वो अपने आप में ऐसे मुड़े हुए हैं. Okay, so you can see how the entire structure is there. Okay, how they are intertwined with each other. Okay, and there are some base pairing, so we are going to study about it, no doubt. But for now, just have a look at the structure. Okay, I will discuss each and everything detailing. Just have a look at the structure. So this is what called double helical structure. So helix means like this, spring-like structure. Okay, helix means like this. This is the helix. Okay, this is the helix. Okay, and there are two helix joined to each other, intertwined with each other, and hence it is called what? Hence it is called double helical structure. Okay. Now have a look. If I'm saying DNA, so what are the constituents? You can say pentose sugar, phosphate group, nitrogenous bases. Okay, pentose sugar. We have already discussed about it. Pentose sugar. Pentose means pentagon. Okay, five sides are there. There is a phosphate group. Nitrogenous bases are there. Nitrogenous bases are further classified into two types on the basis of their structure, purine and pyrimidine. So purine, under purine, what are coming? Adenine and guanine. Under pyrimidine, what are coming? Cytosine and thiamine. Okay, and there is one more structure, that is uracil, coming under pyrimidine only. But I have not mentioned uracil here because uracil is not present in DNA. Uracil is a nitrogenous base which is present in case of RNA. It is not present in DNA. And what is the reason behind it? You are going to study about it in your higher class. Okay, for now, just focus on this particular thing, that DNA, what are the constituent of DNA? So DNA is made up of what? You can see, no doubt, it's made up of nucleotide. Okay, nucleotide is made up of what? So these are the things. Just have a look. We are going to discuss what is nucleotide, what is nucleoside. These are some of the terms that you need to know. So DNA, there is a pentose sugar, there is a phosphate group, there is a nitrogenous base. Nitrogenous base are further classified into two, purine and pyrimidine. Under purine, what are coming? Adenine and guanine. Under pyrimidine, what are coming? Cytosine and thiamine. Okay. Now, there are some terms, nucleoside, nucleotide. What does it mean? Okay, what is nucleotide? What is nucleoside? Okay, so let us have a look at it. Everything is written here. Just have a look. Just have a look. So if there is only sugar, if sugar is attached to nitrogenous base, so what are the nitrogenous base? We have already discussed about it. If you want to see the structure, have a look. So these are the nitrogenous base. This is adenine, this is thymine, this is guanine, this is cytosine. So these are what? These are nitrogenous bases. Okay. And you can see this is what? This is the pentose sugar. This is the pentose sugar. You can count the side 1, 2, 3, 4, 5. So this is what? This is pentose sugar. So pentose sugar is connected to nitrogenous bases. Okay, nitrogenous base. And the bond between them is what kind of bond? The bond is glycosidic bond. Okay, glycosidic bond. Nitrogenous base connected to pentose sugar. The bond between them is glycosidic bond. So this bond is glycosidic bond. You have to remember the bond. These are very important. So this bond is glycosidic bond. Okay. And this whole structure, this pentose sugar and nitrogenous base, this structure is what called nucleoside. Nucleoside. You can see I have written it here. This structure, sugar plus nitrogenous base, how they are connected by glycosidic bond, and this whole structure is called what? This structure is called nucleoside. Okay, don't get confused. There is a difference between nucleoside and nucleotide. And what are the differences? Nucleoside means sugar and nitrogenous base, and nucleotide means what? When a phosphate group is attached to the nucleoside. When a phosphate group is attached to the nucleoside, then it is called nucleotide. And how the phosphate group is attached to the nucleoside? What kind of bond they have? They are having phosphoester bond. They are having phosphoester bond. So you can see here. So this nitrogenous base is connected to this pentose sugar. The bond is glycosidic bond, and this whole structure is called what? This whole structure is called nucleoside. And when a phosphate group is attached, this whole structure, okay, this whole structure. Okay, this C is also coming under this. Okay, and this one, don't get confused. This one is O. Okay, this part is O. If you want to see the structure, let me show you. See, this is the structure of pentose sugar. Okay, so here there is a O. Okay, and there are 1, 2, 3, 4, 5 carbon. Okay, so you can see this whole structure is called what? Nucleoside. And when the nucleoside is connected to phosphate group, then it is called nucleotide. And how the nucleoside is connected to phosphate group? The nucleoside is connected to phosphate group by, there is a bond. What kind of bond it is? The bond is phosphoester bond. Okay, so this bond is what? This bond is phosphoester bond. This bond I'm talking about. Okay, this bond is phosphoester bond. You can see this. So this whole structure, this structure is what? This structure is nucleoside. Okay, and when a phosphate is attached to the nucleoside, and the bond between them is called what? Phosphoester bond. This whole structure is called what? This whole structure is called nucleotide. Nucleotide. Okay, I hope it is clear, all of you. So when one nucleotide is connected to another nucleotide, this is another nucleotide. This is another nucleotide. When one nucleotide is connected to another nucleotide, in that case, the bond is called what? What is the bond they are having? This bond. I'm talking about this bond. Can you see this? This bond. So this bond is what? This bond is called phosphodiester bond. Phosphodiester bond. Okay, I hope it is clear. Let me tell you once again. Yeah, so you can see, what is this bond? This bond is glycosidic bond. This bond is glycosidic bond. Okay, now what are this bond? This bond is phosphoester bond. Phosphoester bond. Okay, now what are this bond? What are this bond? This bond is phosphodiester bond. Phosphodiester bond. Okay, I hope it is clear, all of you. जो मैंने ग्रीन में मार्क किया है वो है ग्लाइकोसाइडिक बॉन्ड. ग्लाइकोसाइडिक बॉन्ड किसके किसके बीच में होता है? नाइट्रोजनस बेस एंड पेंटोज शुगर के बीच में जो बॉन्ड होता है वो होता है ग्लाइकोसाइडिक बॉन्ड। अभी ये जो न्यूक्लियोसाइड और फॉस्फेट के बीच में जो बॉन्ड है, जो रेड कलर में मैंने मार्क किया है, वो है फॉस्फोएस्टर बॉन्ड। एक न्यूक्लियोटाइड और एक न्यूक्लियोटाइड, उन दोनों के बीच में जो बॉन्ड होता है उसे बोलते हैं फॉस्फो diester bond. Okay, so this red one is phosphoester bond, this one is phosphodiester bond, and this one is what? This one is glycosidic bond. Okay, I hope it is clear. I hope this whole thing is clear, everyone. And now you can also mark one more thing here. One more thing. This one is what? This one is, okay, let me tell you. See, just have a look. Just have a look. Okay, so why it is called deoxyribose? In the first slide only, I have already told you what is the difference between DNA and RNA. Okay, so DNA is deoxyribose. Deoxyribose means what? Deoxy means no oxygen or devoid of oxygen. Can you see any difference between these two structure? Can you find any kind of difference? Can you look at this? So these are called what? 1 prime, 2 prime, 3 prime, 4 prime, 5 prime. Okay, so this symbol 1 prime, 2 prime, 3 prime, 4 prime, 5 prime. Okay, you can see at 2 prime position, here there is only hydrogen, there is no O, there is no oxygen, but here oxygen is there, here oxygen is there. So in case of RNA, during bond formation, what kind of bond I'm talking about? Phosphodiester bond, the bond between two nucleotides. Okay, so in case of RNA, this 2 prime can also participate and this 3 prime can also participate. But in case of DNA, only the 3 prime can participate because there is no, in 2 prime, there is no oxygen. In 2 prime, there is no oxygen, so only 3 prime can participate. Okay, I hope it is clear. So in DNA, that phosphodiester bond, the bond between two nucleotides, is formed between what and what? You can see in DNA at the 5 prime end, a phosphate moiety or a phosphate group is present, while in 3 prime end or 3 prime OH is present. So while in RNA, an additional OH is present at the 2 prime end. So I have already discussed about it. There is nothing new written here. So the bond is formed between what and what? 5 prime phosphate and 3 prime OH. In DNA, the bond, the phosphodiester bond, is formed between 5 prime phosphate and 3 prime OH. If you can look at the structure, you can see the bond is formed between what and what? 5 prime phosphate and 3 prime OH. 3 prime OH. Okay. I hope it is clear, all of you. Now let us move to the next part. So this is all about the structure of DNA. So this is all about the structure of DNA. You can see this pentose sugar, there is a phosphate group, there is a nitrogenous base. What are the nitrogenous base? Nitrogenous base are further classified into two, purine and pyrimidine. So what are purine? Adenine and guanine, and which are marked in orange color, you can see. And this thymine and cytosine, these are pyrimidine. And how they are connected, you can see nitrogenous base is connected to pentose sugar by glycosidic bond, and this pentose sugar is connected to phosphate. This whole thing, not only pentose sugar, this nucleoside is connected to phosphate by this phosphoester bond, and one nucleotide is connected to another nucleotide by what kind of bond? That bond is called phosphodiester bond. I hope it is clear what are nucleoside, what are nucleotide. You can see sugar and nitrogenous base, nucleoside, and what kind of bond they are having? Glycosidic bond. Nucleoside, when a phosphate is added to the nucleoside, it is called nucleotide. And what kind of bond they are having? Phosphoester bond. Nucleotide, when one nucleotide is connected to another nucleotide, it is called dinucleotide. And what kind of bond they are having? Phosphodiester bond. More than two nucleotides connected forming polynucleotide. Okay, when one nucleotide is connected to another nucleotide, it is called dinucleotide. When more than two nucleotides are connected to each other, then it is called what? The whole structure is called polynucleotide. And this polynucleotide is what? It is a single-stranded DNA. DNA can exist in two forms. It can exist in the form of single-stranded, means there is only one strand, and it can also exist in the form of double-stranded, means there are two strands. Okay. And why the double helical structure is so famous? Why they have not given any structure like single helix or single helical structure? Because this double helical structure, it is more stable in comparison to the single stranded one. DNA no doubt can exist in both forms. It can exist in the form of single stranded as well as double stranded. But the double stranded one is more stable. Hence, they gave this double stranded or double helical structure. Okay, I hope it is clear. These are very important. Nucleoside, what is nucleotide, what is dinucleotide, and when more than two nucleotides connected, they form a polynucleotide. And when two polynucleotides connected with each other by base pairing, they are called double-stranded DNA. They are called double-stranded DNA. So just have a look. This is the structure of a double-stranded DNA. You can see this is one polynucleotide, this is another polynucleotide. Two polynucleotides, they are connected with each other by this base pairing. Okay, this base pairing. And what are this bond? These are the bond between. These are the bond between what? Can you see it? Between purine and pyrimidine. These are the bond between purine and pyrimidine. As you can see, A is connected to T. And how many bonds they have in between? There are only two hydrogen bonds. And here G is connected to C. And how many bonds they have in between? Three hydrogen bonds. So if someone will ask you, if I want to break the bond, okay, which bond I can break in a more easier way? If someone will ask you, I want to break the bond between this nitrogenous bases. So which bond, like which bond I can break in a more easier way or in a in more faster way? Answer is very simple. You can see the bonding between A and T and the bonding between G and C. So A and T, they are connected with each other by only two hydrogen bonds. G and C, they are connected with each other by three hydrogen bonds. So the answer is very easy, that it will be difficult for us to break the bond between G and C in comparison to A and T. Okay? आपके दो दोस्त थे, एक के साथ आपका bonding is like 60% और एक के साथ no doubt you are bonded, but your bonding is like 40%. Okay? So if someone will try to break the bond, then it will be easier for that particular person to break the bond with that person with whom you have only 40% bonding, जिसके साथ आपका कनेक्टिविटी और आपका जो कंपैटिबिलिटी, आपका जो बॉन्डिंग है 40% है. सो वो बॉन्ड को ब्रेक करना ज्यादा इजी होगा इन कंपैरिजन टू जिसके साथ आपका बॉन्डिंग 60% है. Okay, so the same case is applicable here also. So in order to break the bond between G and C, it is quite difficult because there are three hydrogen bonds. But in order to break the bond between A and T, least amount of energy is needed, and we can easily break the bond. No doubt, energy is needed in both the cases, but here the energy needed is less. Here, in case of G and C, the energy needed will be more, as there are three hydrogen bonds. Okay, so remember, this is also very important. AT and GC, how they are connected with each other. Always remember, A will always pair with T and G will always pair with C. It is not like that A will pair with G, T will pair with C. No, that is not going to happen. You have to remember that A will always pair with T and G will always pair with C. Okay, or else you can also remember purine will pair with pyrimidine. Okay, so A and G, these are what? These are purine. T and C, these are what? Pyrimidine. And you have to remember, A will always pair with T and G will always pair with C. You cannot say, I just said that purine can pair with pyrimidine. That doesn't mean that A will pair with C. That doesn't mean that G will pair with T. No, they have some compatibility. Okay, in your classroom, like you are not compatible with everyone. You are compatible with only few people, and you just, if someone will give you the option, you will choose them. No, I want to sit with this girl, or I want to sit with this boy. Okay, because I'm more compatible with them. I'm not compatible with others. Okay, so the same thing is happening here. Just remember, just keep it in your mind. A will always pair with T and G will always pair with C. And A is pairing with T by 2 hydrogen bond and G is pairing with C by 3 hydrogen bond. Okay, and there is one more thing that you all need to remember. I hope you can easily guess. Like suppose DNA is what? It's a double-stranded structure. Double-stranded structure is more stable. So if at the time of replication we are going to study about it, if the code for one strand is given, you can easily decide the code for the other strand if you remember this. ओके, इफ यू रिमेम्बर, अगर आपको याद होगा कि ए हमेशा टी के साथ पेयर करेगा और जी हमेशा सी के साथ पेयर करेगा, तो अगर एक स्ट्रैंड का कोड आपको पता होगा तो दूसरे स्ट्रैंड का कोड आप इजीली निकाल सकते हो. Okay, so please remember this one. This is very important, and also remember the base pairing between them. Okay, this is also very important. Okay, so let's just move to the next part. So this is the diagrammatic representation. You can see DNA is not like this arrangement is a hypothetical. Okay, ये जो डायग्राम उन्होंने दिया है ऐसे अरेंजमेंट नहीं होता है. ये जो अरेंजमेंट होता है मोस्टली वो लोग ट्विस्टेड होते हैं. ओके, इंटरवाइंड होते हैं. एज यू कैन सी ऐसे होता है स्ट्रक्चर. हेंस इट इज कॉल्ड डबल हेलिकल स्ट्रक्चर. ओके, हेलिक्स के तरह होते हैं. और कुछ टर्न्स होता है. यू कैन सी दिस आर द टर्न्स आई एम टॉकिंग अबाउट. दिस आर द टर्न्स. यहाँ पे एक टर्न है, यहाँ पे एक टर्न है, यहाँ पे एक टर्न है. सो दिस आर द टर्न. In this turn there are some base pair. There are some base pair. So now I think all of you can understand what are base pair. Base pair means A will pair with T and G will pair with C. Okay, A will pair with T with the help of 2 hydrogen bond and G will pair with C with the help of 3 hydrogen bond. So these are what called base pair. So ये जो DNA होता है वो ऐसे straight नहीं होता, intertwined होता है. So there are some turning, there are some turn, there are some point where they are turned. Okay, so एक टर्न में कितना बेस पेयर है, एक टर्न में कितने बेस पेयर आते हैं. ऑन दैट बेसिस we can tell what is the pitch. Okay, so this is also very important. Okay, in most of your medical entrance examination you can expect they will give you the distance between two base pair and they will give the number of base pair present in one turn and they will ask you to find the pitch. Okay, so what is this? So these two polynucleotides are connected in a right-handed fashion. What do you mean by right-handed? Okay, like in a right-handed way, not in a left-handed. The turning, जो turning हुआ है, वो right-handed way में हुआ है. Right-handed way में हुआ है. So these two polynucleotides are connected in a right-handed fashion. Each turn consists of 10 base pair. If I'm talking about the turn, you can see these are the turn. So यहाँ पे picture में इतना अच्छे से नहीं दिखाए हैं वो लोग, but यहाँ पे 1, 2, 3, 4 ऐसे काउंट करोगे तो यहाँ पे 10 बेस पेयर रहेंगे. Each turn में 10 बेस पेयर रहेंगे. For example, if I'm saying, let me draw here 1, 2. Okay, so I'm just gonna count 1. This is one base pair. 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 1, 2, 3, 4. Then 5. Then 6. Then 7. Then 8. Then 9. Then 10. तो यहाँ पे एक बार टर्न करेगा, फिर और एक बार 10 बेस पेयर आ जाएंगे, उसके बाद और एक बार टर्न करेगा, फिर 10 बेस पेयर आएंगे, उसके बाद और एक बार टर्न करेगा. सो इन वन टर्न देयर आर हाउ मेनी बेस पेयर? इन वन टर्न देयर आर 10 बेस पेयर. और दो बेस पेयर के बीच में डिस्टेंस क्या है? इफ आई एम सेइंग द डिस्टेंस बिटवीन टू बेस पेयर, मतलब ये. इफ आई एम टॉकिंग अबाउट द डिस्टेंस बिटवीन टू बेस पेयर, मतलब आई एम टॉकिंग अबाउट दिस. ये एक बेस पेयर है, ये एक बेस पेयर है. तो ये जो बेस पेयर के बीच में जो डिस्टेंस है, ये जो डिस्टेंस है, ये जो दो बेस पेयर के बीच में ये जो डिस्टेंस है वो कितना है? व्हाट इज द डिस्टेंस बिटवीन दिस टू बेस पेयर? यू कैन सी here it is written, it is 0.34 nanometer. 0.34 nanometer. Nanometer means what? 0.34 into 10 to the power minus 9. Okay, so the distance between two base pair is 0.34 nanometer. So now we can easily find out the pitch. So what is pitch? It is the distance between two base pair and number of base pair present in one turn. So here it is written, what is the distance between two base pair? The distance between two base pair is 0.34 nanometer into number of base pair per turn. In one turn, how many base pair are there? 10 base pair are there. So all you need to do is 0.34 into 10. 0.34 into 10. So what will be the pitch? Pitch will be 3.4. It will be 3.4. Okay. And if someone will ask you, what is the length of polynucleotide? You can see distance between two consecutive nucleotide. Total number of base pair present in the polynucleotide. Okay, how many base pairs are there in the whole polynucleotide? How many base pair are there? You have to find it out. And what is the distance between two consecutive nucleotide? Okay, what is the distance between two consecutive nucleotide? In this way, you can find the length of the polynucleotide. So these are the two formula that you need to keep in your mind. One is what is the pitch and what is the length of the polynucleotide. Okay, and there is another rule called Chargaff rule. Okay, there is another rule called Chargaff rule. So according to them, the ratio between A and T and G and C are constant and equal. Okay, the ratio between A and T and G and C are constant and equal. That is A by T is equal to G by C. And A, purine, per pyrimidine of purine divided by pyrimidine is 1. The ratio of purine by pyrimidine is 1. Okay, the ratio of purine and pyrimidine is 1. So what are purine? Adenine and guanine. What are pyrimidine? Cytosine and thymine. So A plus G by T plus C is equal to 1. So you can remember this formula, this rule in either of this way. You can remember the ratio between purine and pyrimidine should be 1. Okay, and you know what are the purine? It is adenine and guanine. And you also know what are the pyrimidine? It is thymine and cytosine. So you can easily remember in this way. Just remember the ratio between A and T and G and C are constant and equal. Okay, you can remember it in a very simple way. You know that A will always pair with T and G will always pair with C. Okay, and you also know that in order, like if you want the two strands to join with each other, okay, if you want the two strands to join with each other, base pairing is needed. Base pairing is needed. दो स्ट्रैंड आपस में ज्वाइन होने के लिए बेस पेयरिंग जरूरी है. और बेस पेयरिंग किसके किसके बीच में होगा? ए हमेशा टी के साथ पेयर करेगा, जी हमेशा सी के साथ पेयर करेगा. तो डीएनए में जितना ए होगा उतना टी होगा, नो डाउट, बिकॉज ए विल ऑलवेज पेयर विथ टी. एंड जितना जी होगा उतना सी होगा, बिकॉज G will always pair with C only. Okay. So accordingly you can remember. We are going to solve some question on this particular rule, Chargaff rule. But you just have to remember, you just have to keep only one thing in your mind, that A will always pair with T and G will always pair with C. अगर A 4 है तो T भी 4 होंगे. अगर G 5 है तो C भी 5 होंगे. क्योंकि पेयरिंग के लिए उन्हें जोड़े में रहना जरूरी है. Okay. For example, there are 5G, but there are only 4C. What will happen to the other G? The one G will remain single, right? Base pairing जरूरी है डबल स्ट्रैंडेड स्ट्रक्चर फॉर्म होने के लिए और डबल हेलिकल स्ट्रक्चर फॉर्म होने के लिए. So you just have to keep this thing in your mind and also remember this one, purine by pyrimidine is equal to 1. So you know what is purine, you know what is pyrimidine. So the ratio between A and T and G and C are constant and equal. Okay, this entire rule will be more clearer to you when I will solve the question on this rule. And this is very important, no doubt this is very important. This entire slide is very important. Okay, so what is the formula for pH, what is the formula for length of polynucleotide, and what is Chargaff rule. So this entire slide is very important. Now just have a look. So these are your textbook question. If the length of E. coli DNA is 1.36 mm, can you calculate the number of base pair in E. coli? I have already provided you the formula for finding the base pair if length is given. If length is given, how to find out the base pair. And if base pair is given, how to find out the length. I have already provided you the formula for it. And group the following as nitrogenous bases and nucleoside. There is another question. Okay, there is another question. Yeah, just have a look how to distinguish which one is nucleoside and which one is nitrogenous base. So what are the nitrogenous base that we have did? Adenine, guanine, uracil, these all are what? These all are nitrogenous bases. And what is nucleoside? Nucleoside means the nitrogenous base is connected to sugar. So when adenine is connected to sugar, what is the name? Adenosine. Adenosine. In case of RNA. In case of DNA, it will be deoxyadenosine. And guanine is also a nitrogenous base. It is a nitrogenous base, but when sugar is added to it, the name will be guanosine in case of RNA. In case of DNA, it will be deoxyguanosine. And if I'm talking about uracil, when sugar is added to it, it will be uridine. So I hope it is clear that if they will give some name like adenosine, guanosine, uridine, adenine, just like this question, you can easily tell me which are nucleoside and which are nitrogenous bases. I hope you can easily solve this question. This is your textbook question only. Adenine, cytidine, thymine, guanosine, uracil, cytosine. You can easily distinguish which are nucleoside and which are nitrogenous bases. So just give it a try. All of you just give it a try at your home. Next class, we are going to solve this question. We are going to continue this entire topic from here only. So I hope it is clear, all of you. I hope the entire thing is clear, whatever you have read. So you just have to keep in your mind what are the things present in DNA and you have to keep in your mind that how they are connected. What is nucleoside? What is nucleotide? This slide is very important. What is nucleoside? What is nucleotide? What is dinucleotide? How how they are connected with each other? What are the bonds? We have already discussed about all these things. Okay, so you have to keep in your mind that A will always pair with T, G will always pair with C, A will always pair with T by how many bonds? The bonds between them are, there are 2 hydrogen bond. G will always pair with C by 3 hydrogen bond. So I hope it is clear, all of you. This is the double-stranded polynucleotide. This is the single-stranded one. This is the single-stranded one. What is the difference between DNA? What is the difference between RNA? So I hope everything is clear. So this much for today's session. Thank you, all of you. And in order to join my channel, you can click on this link. If you like this video, please do like, share and subscribe to my channel. And if you have any doubt or any queries related to this entire topic, what I have completed today, you can comment. Okay. So bye everyone. Take care.